Asperaka.
Okay, let's get going. Welcome to our first Discovery Dialogue session, which is being held live on the second floor amphitheater space in the Peter Gilgan Centre for Research and Learning at the Hospital for Sick Children. And today we have Dr. Jayne Danska and Dr. Michael Taylor joining us. So welcome. Thank you for joining us today. I became Chief of Research at SickKids just about a year ago, and one of the things that is top of my mind for me to do is try to generate the best investments in research to enrich the culture and opportunity for discovery. We have a long history of discovery here at SickKids, but in my initial assessment, there's no simple set of guidelines or rules that emerge that point to what the underlying principles of success might be in the discovery process. As a starting point, a few months back, digging deeper into what these ingredients may be, one thing we did was generate a list of scholarly biomedical publications that originated from scientists here at SickKids that have been cited over 1,000 times. For those of you who may not know, having a paper cited 300 times is considered a citation classic. So 1,000 plus means really a super citation classic. For me, citation statistics like this are most important because it is your peers who, through their citation of your work, really list the priority of the importance of that paper. So when we did this experiment, this exercise, there were originally 25 papers that came from the SickKids list. As you might imagine, the famous cystic fibrosis gene papers, gene discovery papers led by Joanna Romans and Lapchi Choi are at the very top of the list. There are papers from Peter Dirk's team on his brain cancer stem cell discoveries. Mike Salter had work on his pain and the molecules underlying pain. These were at the top of the list also. And some of the work from my own laboratory on copy number variation and autism were on the list. And even more recently, as you heard, just John Dick, who is this year's International Gairdner Award winner. For those of you who don't remember, he performed his early research here at SickKids and his contributions to the discovery of cancer stem cells. Those papers are right at the top of the list also. And there are many others too, but there were 25 on the initial list. And just recently, in the last few months, Jayne and Michael's papers crossed the threshold to move into the 1,000 range. So I wanted to use this time to hear their stories, what led to this success, and see if we can learn some of the things that have worked for them over their career. So to begin with, I just wanted to introduce them and actually have them introduce themselves. So I'll invite you, Jayne, and then Michael, to just give us a little bit of background, where you're from, what your academic pathway was, and how you landed here at SickKids. And Jayne, if you can go first, please.
Sure. Well, thanks for doing this, Stephen, and having Michael and me here. I'm from New York City. I was actually, a little known fact, I was born on the 59th Street Bridge, going over the East River. So born and raised in New York, did all of my education in the US, went to a great small liberal arts college as an undergrad, studied biology, politics, religion, all kinds of things, then to Cornell University Medical School in New York City, where I did a PhD there and with research at the Cold Spring Harbor Labs in molecular biology, molecular genetics. And then I went to Stanford University Medical School in California, where I started to study immunology, and that has been the field that has absorbed my interest over the many years. I came here to SickKids from Stanford at the end of my postdoc, having looked at jobs in many large US cities as well, and concluding that Toronto was a place that made sense to a New Yorker, and having lots of support from my Stanford mentors about the collegiality and productivity of SickKids, about which I didn't know too much at the time. So I have been here ever since. I'm now in the program in genetics and genome biology, and at the university appointed in departments of immunology and medical biophysics.
Wow, that's a great story, and maybe we'll get back to the 59th Street Bridge a little later.
That's my mom's story, yeah.
Yeah, Michael, go ahead.
I was born in Calgary, Alberta. I'm a prairie boy, although I've lived all over the country. I went to undergrad and medical school at the University of Western Ontario after high school because I heard that it was a fun place to go to school, and it was not wrong. It was also an easy place to get high marks, so I could get into medical school. During medical school, I found out that medicine was not science, and I wanted to keep going in science. So I decided after consultation with my wife, and we came back to Toronto, where I did a neurosurgery residency here in Toronto, and combined that with a PhD in Jim Rucka's lab in the old McMaster building, where the new building is now going up. After that, I did a postdoctoral fellowship at St. Jude's Children's Research Hospital in the United States with Richard Gilbertson in developmental biology, as well as a fellowship in pediatric brain tumor surgery with Alex Sanford and Rick Boop. Then when I came home, I did a second postdoc with Janet Ressant in functional genomics, and I think I was supposed to be with her for longer, but after about 2 years, she took the big job over here, and so she told me I had to get out of her lab and start my own. Throughout my entire career, I've been studying these two brain tumors, medulloblastoma and ependymoma, because there's something that I interact with in my clinical practice and that I think I understand very well. And they're clearly unresolved problems. And they're also very rich in developmental biology, which has been one of my loves.
Great. Okay, so let's go back to this concept of 1,000+ citation papers. And Jayne, can you tell us a little bit about your paper that was published in Science? And the title of that paper is Sex Differences in the Gut Microbiome Drive Hormone-Dependent Regulation of Autoimmunity. From your perspective, what made this paper different from all the other ones in your laboratory that made it your highest impact paper?
I've always been interested in biological sex as a variable in determining gene expression, in determining both normal development and pathology. And nowhere is this more of an issue than in autoimmune disease, which are far more frequent in females than in males. And it always struck me that despite all of our amazing advances in genome-wide association studies, where we could collect, in the case of type 1 diabetes or lupus, hundreds of markers of disease risk, even collectively, for lupus, for example, if you inherited all of the known variants of risk, they don't collectively add up to the additional risk of being a female, because the disease affects 10 times as many females as males. And there was so little understood about, and remains so little understood about that biology. So this paper was working in a mouse model of type 1 diabetes. And in this mouse model, there's a higher proportion of females that succumb to the disease, a spontaneous disease, than males. And I'd been studying the genetics of that for a long time, identified a few genetic loci in the mouse model that had different effects in the male and female mice. And so we had been on that, kind of sniffing on that trail for a while at the genetic angle. But it was when we moved those mice into a fully germ-free environment to try to understand how environmental hygiene affected the disease process that we recognized that the sex effect was totally determined by colonization with microbes, because in a germ-free environment, the males and the females got equal disease frequencies. So I thought it was all genetics, but now I appreciated that it was really a major environmental trigger. So that paper set upon trying to understand the rules of how that worked, transferring microbes from male mice into females and so forth, and being able to show through an androgen-dependent mechanism that the microbes that inhabit a male are distinct from those of a female, at least in aspects, and that those are being modeled by and are modeling the hormone patterns in those animals, which in turn was affecting this autoimmune disease. So that hadn't been seen before, and is certainly not by us. So
yeah, so I'll come to Michael's in a second, but so I remember when the paper came out, and I knew about your science. I was surprised that you actually published in this field. I remember I sent you an email. So this is great. And what was it? What was the combination of factors that gave you the confidence to submit this to Science in one of the top leading journals? Was it the topic or the results, or just the story at the right time? What was that?
Yeah, it was the long-term interest in sex effects and really still lacking good mechanistic information on how they took place, and then having this black and white result with the transfer of the male microbes into the female mice. It was such a spectacular difference, far better than any of the genetic mapping I'd ever done in these complex traits, that it just seemed that it would be impactful, and it wasn't a terrible ride getting it through at Science.
Yeah. I did a similar session like this with TAC-MAC many years ago, and David Naylor was the chair, and we ended up talking a lot about the importance of the title and the abstract to get your message across and how it plays into actually citations. And I remember, Jayne, when we told her she made the 1,000+, she said having sex in the title was the key.
Not a bad plan when it fits.
Yeah. Anyways, okay. This is a great paper. So that paper, people should look at it. It's one of the really first papers I saw on microbiome that really caught my attention. So, Michael, your paper, the title of your paper that made the list is Molecular Subgroups of Medulloblastoma, the Current Consensus, and this was published in Acta Neuropathology. My interpretation is it's a culmination of data from your early discovery papers, and you can talk about that paper, but I wanted to know what you thought your most important discovery paper was that kind of led to that, and tell us a little bit about that process. So I just want to say he's got several Nature papers that are going to cross the threshold sometime soon, but tell us which one you think is the most important.
Well, that ACTA paper is really perhaps more a demonstration of chutzpah than anything else, to be honest, because it's almost, I wouldn't call it a review paper, but around the time when a lot of us were agitating and starting to use molecular biology rather than human retinas to classify childhood brain tumors, we had met and we'd had tried to have a consensus conference about how we were going to go forward and how we were going to use these molecular diagnostic tools and the molecular information as opposed to, again, human retinas and human occipital association cortices. And after the meeting, at the meeting, feelings were high. And then we went home, everyone's like, oh, but maybe we should think about this for another little while. Why don't we debate it until we're all just blue in the face? So I just wrote it up and submitted the paper. And then it got accepted, and I made sure that everybody was an author, and as a result, it was a consensus by everybody who was an author.
All right.
The data that paper came out of was principally driven, at least from my lab, from a JCO paper, Journal of Clinical Oncology paper, that I think has about 1,300 citations right now on Google Scholar. And what we did in that was, in the old days, I mean like when I was a resident, the way that they would diagnose a brain tumor was a surgeon would chop a piece of it out, and then somebody would walk it down the hall to the pathologist, and the pathologist would cut really thin slices of it like it was prosciutto, and then they dump some ink on it and look at it under the microscope. And that's the same way they used to do it in like 1910. Yeah. And we started using molecular biology instead. And what we noticed was that whereas there were groups of brain tumors that looked very similar, or at least couldn't be sorted out from each other using human retinas and occipital association cortices. You know what I mean, right? I'm trying to make fun of the idea that, we're using humans and brains as opposed to computers. But once we started using genomics and allowing the genomics to agnostically tell us what was there, a lot of the heterogeneity just sorted itself out very clearly. Yeah. There were quite a few people who were very uncomfortable with that, right? You've been on committee meetings where I've been there too, where, the old dogma was one postdoc, one protein, one paper. And the idea to do science was you would read a bunch of stuff and you'd talk to a bunch of people, and you'd have an idea and you'd make a hypothesis and you'd go out and falsify it. And then when genomics came on the scene, the idea was, well, I'm interested in this, so I'm going to throw genomics at it, beat it with a hammer, see what falls out, pick that, and then try to falsify that. Right. And that has been a very successful recipe for a number of us. Yeah. And that allowed us to tell the difference between these different types of medulloblastomas that look the same under the microscope, or at least similar. But once we look at them molecularly, they're very different diseases.
And which one of your studies was the one that gave you, the key insight, clue that said you could actually do this?
I think it was incremental. Yeah. But the one that would probably, if you had to place a pin on it somewhere, would be Paul Northcott's Journal of Clinical Oncology paper. Oh. And there were a number of other people, in the field at the time, like Richard Gilbertson and Marcel Kuhl, Scott Pomeroy, Stephen Pfister, and we were comparing results, and we didn't see exactly the same things, but we were seeing very similar things. And it was clear to all of us at that time that the days of the optical microscope were numbered.
Right. And what year was that? I'll come back to this later.
Probably right around the time of probably 2010, I don't know.
2000, yeah, okay. Okay, great. So back to Jayne. Jayne, you've had several successes in your career, and most recently there was a commercialization success that spun out of research you did many years ago in collaboration with John Dick, whose name I mentioned earlier. Can you tell us about how this came about and why it was important and where it's going to go from here?
I mean, that's an amazing sort of arc of the story. It goes really the whole cycle of research. It started with, gee whiz, I wonder how this works, which I think is often what drives many of us as scientists. We like to solve puzzles and understand mechanism. John Dick had been, as you alluded to, he'd identified stem cell biology and hierarchies in humans from normal blood stem cells, as well as later from blood cancer stem cells. And he'd done it in large part by being able to transfer the human progenitor cells into permissive mouse models, immune deficient mouse models. And they'd seen over some period of time that there were some immune deficient mouse models on particular strain backgrounds that were permissive, that would allow the human cells to engraft. But if you put those same immune deficiency mutations on other genetic backgrounds of mice, they didn't engraft at all. So to a geneticist, of course, this smells very strongly of a genetic control mechanism. And we'd been working in the context of the diabetes work that I mentioned earlier. We'd been working on slicing and dicing the genome of this diabetes-prone mouse strain, and so had essentially a ranch of different so-called congenic strains of these mice. And this was the strain of mouse that was permissive for human engraftment. So for a couple of years, I'd been saying to John, over coffee and best conversations over beer, of course, to say, we really should try to solve this using genetics. And, we maybe have a great toolbox for doing it. So eventually he had a postdoc who was, crazy and keen, the two combination factors that we always need, or at least brave and keen. And so we started working on this together using the agnostic genetic approach to try to identify the gene that was responsible for engraftment or not. Found it. Turned out to be an innate immune recognition system molecule that's polymorphic in humans, polymorphic in mice. And we recognized that this might be a system that we could exploit in the way that the standard T-cell immune checkpoint inhibitors are now working. But on the innate immune side, this is macrophages as the active cells here, that we might be able to develop a new kind of medicine that would work in that axis, that would provoke or allow macrophages to recognize and kill cancer cells, particularly blood cancer cells, is what we started with. Turned out to work. We developed the beta version here at SickKids and made the proteins in my lab when we were at the Mars building. Mm-hmm. And we partnered with John Dick and myself and Jean Wang, also at UHN, who was the third important contributor. We partnered with a small biotech company that had really been spun out of the U of T, and by that point was running on the sort of fumes that our biotech companies often do. We got some money through Genome Canada. I'm very glad that they invested in us. We really needed that opportunity. And then eventually the company was able to build it into a prospect. We built enough value that other investors were interested. It went to phase 1 clinical trials. It had very favorable low toxicity profile. And we even saw some signals of remission in a mixed bag of blood cancers. And as you know, phase 1 trial patients are really quite ill and often have been refractory to multiple standard treatments. So over, I guess, about 4 years of the clinical trials being run through Trillium Technology Inc. here in the Toronto area, it caught the attention of some big players. And last year, Pfizer acquired the company to acquire the license for our drug, which SickKids holds and UHN holds. And hopefully now it's really going to go to phase 3 trials. So it really went the whole way from, gee whiz, this is a cool puzzle, let's solve it with genetics, to identifying a new drug.
And what was the timeline for that from primary discovery to...
So the discovery was in like 2007-8. The application to showing that it might work against human primary cancer cells, and we got those cancer cells from our biobanks here at SickKids. And that was about 3 years later. So by 2012, we knew that it was going to work, at least in theory. And by 2021, the picture took place. So
wow.
10 years, 12 years.
Okay. We got a little background noise up there. We are live. That's okay. Okay, so that's an incredible story. Michael, so 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 you're a brain surgeon, an MD, PhD, as you told us. How the heck do you have time to run a world-leading molecular biology lab? And I remember we took the subway home just about a week ago, and you told me on the way home that you thought maybe 10 to 20 years out, you might be able to cure some types, some of the subtypes. So play back to your top-cited paper of medulloblastoma. What's the story there?
My schedule. First of all, I love deadlines. I love watching them whoosh past. They make such a pretty noise. To paraphrase one of my favorite authors, Douglas Adams. And I learned over the years to be very good at delegating. I have a great team who works with me, Xiao Chong and Craig and Mira, and the four of us together can get just about anything done. I really enjoy having my autonomy respected, and I try to make sure that everybody in my lab has a lot of freedom and autonomy too. And when I give my people their autonomy and their freedom, quite often they shine. And I think by doing that, I've been able to run a lab and still maintain my admittedly scaled down practice. What we're talking about on the subway is rather than actually curing some of the kinds of medulloblastoma, I think that maybe we could prevent them, which is kind of funny because the Canadian Cancer Society for years has been harping on about preventing cancer and not curing cancer, and it used to annoy the hell out of me because I thought it really had no application to childhood cancer, and I thought, as usual, everyone was ignoring pediatric cancer. But recently it's come to light that human brain development, particularly the human hindbrain and human cerebellar development, is very different from the mouse. And after some work that was done by Parthiv Haldapur and Kathy Millen in Seattle on normal hindbrain and cerebellar development, we realized how it was related to human medulloblastoma, because human medulloblastoma arises in the hindbrain. For decades now, moms have been asking me, when did this start and why did this start? And for an average group 4 medulloblastoma, which is the most common type, the kid's usually about 8 years old, and I'd have to say, I don't know and I don't know. Probably just bad luck, right? Cells are dividing, there's errors, and it's, that's the way it goes. But now we've actually found out, we think, when exactly these tumors start using single cell technologies, because there are types of cells that are present in the tumor and that are present early in development and nowhere in between.
Right.
And most of us in the field don't believe that any of the cells dedifferentiate. So we think now that the group 3 and the group 4 tumors start either in the first trimester at the border between the first and the second trimester of human development, which is kind of fascinating. So what does that have to do with preventing things? So we prevent colon cancer in our society by looking for adenomas, polyps. It's a sort of a pre-malignant, partially transformed clone that's in the colon, and you take it out before it becomes full-fledged colon cancer. Because taking out a polyp is easy, and when you do it, it's done. When you take out a colon cancer after there's mets in the liver and the lungs and stuff, it's too late.
Right.
So by understanding the molecular biology of the disease and relating it back to these very early events in human development, we've now shown that there is the version of a polyp that exists in the hindbrain of humans that does not appear to exist in the hindbrain of other species, including macaques. We haven't checked chimps or bonobos yet. And that for some reason, human cerebellums are huge compared to almost every other species. What percentage of you're a neuroscientist, I shouldn't put you on the spot, especially since you're my boss, but what percentage of your neurons do you think are in your cerebellum? Most of us would have thought not very many, right? Because your cerebellum doesn't do poetry or calculus or any of the things we think are so human and impressive. But in fact, 80% of the neurons in your brain are in your cerebellum, right? And because of this blowing up of the cerebellum, that's why humans get medulloblastoma. It's part of the human condition. And this little tiny area in a very specific portion of the cerebellum called the nodulus is where all these cells stream out of over development.
Right.
It's got to make 80% or 69 billion of the neurons in your brain. For most of us, by the time we're born, that area is done. It's a desert. Everybody's divided and they've moved out. But in a very small percentage of people, there's still some undifferentiated cells there. Either stem cells or progenitor cells, usually due to mutations, and we found the mutations now.
Yeah.
And those just slowly grow over time. That's present at birth. The kids don't really get full-fledged medulloblastoma until they're 8. That gives me 7 years to find those kids and destroy that nodulus. Right. And I'm starting to work with a number of people now so we can figure out how to do this. I think it's actually more feasible probably to prevent this across our species than it is to cure it, because we won't have to deal with the genetic heterogeneity that arises over the course of the cancer's life.
So when you said it would take 10 to 20 years, why so long? Why can't you
I
just
think
start
it's
now?
going to take 10 or 20 years because I'm just going to have to convince people to give me enough money to do it.
Right.
I think if I had $10 billion, I could do it in Toronto tomorrow.
Right.
We just have to have every baby go into the MRI. So I think what's going to take the time is we're going to need to develop some sort of a biochemical or serum test to find oncofetal antigens within the blood or the serum of infants, and then use that to enrich for kids who are at risk. Image those ones, because we can't image everybody yet. Although I do believe that every human baby is going to be imaged within the next 20 years, MR. I'm sure that's going to happen, right? If you don't believe me, have a look at your phone. Remember what your phone looked like 20 years ago.
Yeah.
And then in the kids who have it, a surgeon will go in and take it out, or someone will stick a wire in it and zap it, or my friend Dwayne Mitchell in Florida will make a vaccine. But we're going to prevent it rather than cure it.
So coming back to the opening comments, you mentioned the people you worked with and who help you with deadlines, do the science trainees, and Jayne also, in addition to be a senior scientist, is the associate chief of faculty, and you mentioned your collaborators. Tell me a little bit more about what your secret to success has been. What's different here? Why are these discoveries happening here? What do we need to invest more in?
One that actually, I haven't talked to you about this in a long time, Michael, but years ago we were studying a mouse model of leukemia. I also work on leukemia. And the mouse model was of particular interest because the leukemia invaded the central nervous system at high frequency. And we asked Michael's help in evaluating the histology. I don't know if you remember this mouse model.
I remember when you came for a beer with my mother-in-law. So.
Oh, that's right. Gosh, I hope I was well behaved. And
You're not the one we had to worry about.
So and you were a co-author on that cancer cell paper. We had, it was nice. And so, map forward now more than a decade again. And I really stayed looking at this issue of how, and you made the point at the time, we don't know how leukemia cells, Michael's spoken of a cell of origin in the brain, but we don't know how leukemia cells enter the CNS, right? That there may probably multiple mechanisms, not well understood. So we've continued to work on that problem and identified at least one new mechanism by which they enter by drilling through the bone, and published that recently. But that preoccupation of mine was definitely triggered by conversations with Michael early on, because he was the expert we brought in to look at the brains of these mice. So for me, I think one of the key things about SickKids is not just that there are all kinds of great creative minds here. They talk to each other.
Right. Yeah. Do you want to comment?
I'd say one of the things that I learned around here is that it's a very big world, and that as a Canadian, we can get along with everybody. The flow through of fellows in neurosurgery at SickKids is pretty spectacular. There's people from just all over the world that have been flowing through, and I was able to meet people from all over the world. And when I started my own genomics efforts, I was reaching out to people from all over the world, some of whom had trained here and others who hadn't, but also trading on our reputation as Canadians, as builders rather than gatherers. And I was able to build large genomic consortia that weren't named after me or named after the institution, but named after the goals of the institution, the goals of the consortia. And because of that, large numbers of people were able to get behind me, to help me, to give me samples. But also because I've always been very inclusive with authorship, there's been a lot less pushback against a lot of my ideas, because most of the people who might have pushed back are authors.
Shared investment.
Shared investment.
What about trainees? There's always a key postdoc, or you mentioned earlier, someone with the fire in their belly to get things done. And
for me, there have been great postdocs too, for sure, but I've had some amazing graduate students. I mean, the first author of the paper that you mentioned earlier on sex effects in the microbiome is a graduate student, came from University of Western Ontario as an undergraduate, incredibly bright woman, worked on these projects with me, published very well, went to Rockefeller as a postdoc, and now is a faculty member at Vanderbilt running her own program. So I've gotten some really amazing graduate students. So I'd have to go there first. I think the students here are terrific.
I'd say the same thing. You know, I've had really, really good graduate students, as well as postdocs from around the world. The Canadian graduate students, the ones that come to Toronto, are usually want to work and they want big papers.
Right.
One of the ones that does stand out, the one I mentioned before, Paul Northcott, who, as first and contributing author during his PhD, had 50 publications. And I've heard other people in the brain tumor field now say, when they're starting their PhD, that they want to pull in Northcott.
Right.
I don't think anyone's done it since.
Well, yeah, I think that's the answer, right? Is people
You were on his committee.
No, I'm a co-author on the paper.
Yeah, you are.
Thank you for
Another example.
including me. Yes.
I'm a Canadian. I put everybody on the paper.
Yeah, he was in Montreal for a while? Or is
No,
he
no, he's faculty at St. Jude now.
Okay.
And has had multiple Nature papers.
Yeah.
He and I have sort of accepted papers right now, even.
So we need to get it back here. But, so the issue of access to technology you mentioned too, but I think for me you set it up perfectly. Is really the successes you emulate those who you train with. You strive towards the same characteristics and successes of the people you were mentored by, and I mentioned a lot of names, and John Dick's name, Lapchi Choi's name. These people are all on the ninth floor. What was it? Ninth floor, 11th floor? I can't remember now. 11th floor of the Elm Wing. And I got to see these my academic heroes when I was a graduate student. I think that's part of it, to have the generations of science coming up and training each other and passing the stories on. And that's part of the reason we're having this dialogue series. On that note, do you have any advice for the next generation of scientists coming in and types of people we should be recruiting, what they should and shouldn't be doing? This is your job, I know, Jayne, as faculty, but I'd like to hear both of your comments and advice.
Yeah, I guess when I think about what are winning formulae for people, and there are many, I don't mean to say it's one only. It has to start with fundamental curiosity and passion about your area of research. If you don't have that, it's going to be hard to do something that's really novel and innovative. But now I think really one of the key issues is being multidisciplinary, is learning to have strengths, not just in genetics or not just in studying the brain, but in being able to think broadly and incorporate new tools and methods, including computational methods, so that you can learn to make use of, big data is just data, it's just information. The keys are going to come into making it into biological knowledge, right, and how that can be done. So I think it's both the curiosity and passion for your area, but also an openness to integrating multiple approaches together and learning from your colleagues to be able to do it.
Michael?
I guess I'd start off by echoing what Jayne said about curiosity and energy, because there are very few people who end up doing very well who lack either one of those two components.
Right.
They're not enough, but they're necessary but not sufficient. One of the things that I have seen, it's not always the right way to do it, but there are certainly people who are very cautious and people who are not cautious. Right now, to become a new PI, which is what a lot of us try to do with our trainees, to spin them off as PIs, you really need at least one paper in a fancy journal. And sometimes you need two nowadays, which is a little bit crazy. And to do that, you can't just ask incremental questions. You have to ask questions where you could be wrong. And if you're, what I keep telling my people all the time is, if you're never ever wrong, you'll never be right in a meaningful way. So we need to open ourselves up to the idea that we can do things that might be wrong as long as we're doing them out of the best of our intentions and with our eyes open. And that sometimes big ideas are necessary, because is the juice worth the squeeze? You should ask yourself before you start the project. It's all published. How excited do you think people are going to be able to read about it if it turns out that everything you thought it was true?
Yeah, so I think that's a great point. And this whole concept of breadth, being a scientist as opposed to a specialist, having the background and the knowledge to come up with the new ideas is really key, right? So do you think that's something that can be learned? And how important is the environment, or is it innate? Is it a mix? You talked about the genomics approach, cast a wide net, generate lots of hypotheses, but you still have to pick the most important one and have a high batting average. You want to just touch on that?
I think environment is extremely important in the point that you've just raised, Steve.
Mm-hmm.
And so having access to the technologies being used at a high level in the context of people that are really shaping, as Michael's alluding to, big questions you probably aren't going to be able to answer, but if you're not trying, then you're not really working at the tip of the spear.
Yeah.
So you won't always penetrate what you're trying to get through, but at least that's where you're really trying to work. And sometimes, the side eddies also move you forward. So you don't always know, but having access to an environment that's intellectually intense and stimulating, collaborative, together with available technologies, I think environment's really important for those features.
Great. Any closing comments?
I think luck. We haven't touched on luck yet. There's always a little bit of luck in these things because we've all had projects that really seemed like they were going in the right direction and they made incremental advances. But you need just a little touch of luck, and that also requires being willing to take a little risk.
And you have to be able to take the beat down. So after you get unlucky 3 times, you can get back up off the floor and do it again and manage to get lucky on the 4th try.
Yeah. I think luck
Resilience.
is maybe it's recognizing opportunity, and if you planted 1,000 seeds, some of them are going to go, but you have to still see that, be able to observe this, and that's really what the discovery process is, right? Making sense of something and seeing it for the first time, and then describing it to the public or your colleagues so they can understand the importance of it.
And knowing you made the discovery.
I've
seen any number of times where people have published high impact papers where the same data went under other people's noses and they thought it was an artifact or they missed it completely.
Yeah.
Well, that was our story in the copy number variation, and I remember exactly where it finally clicked in my head. I was riding my bike home 2003 on college in Ossington. Stopped at a red light. I realized the importance of it. We had it for several months, but didn't realize the importance, and that's the discovery, actually, when you realize the importance. Okay, well, this has been great. We're going to have this type of this discovery dialogue session probably on a quarterly basis, and we're going to keep this in-person setting here in the amphitheater, and I hope you enjoyed the session today. I learned a lot from both Jayne and Michael, got some good ideas, and we'll thank you and see you next time.
Great. Lots of fun.